So what are you up to today?
I'm going to be imaging.
Fun.
Are you up to your boss essays?
Yeah, got another one.
796 long.
Yep. So in this lab, we study how cells communicate. What we're studying specifically is carbon black. That's a particle of air pollution. And when the human lung cells that we're studying take in the carbon black particles, and as the particles accumulate, what signals are being induced in these cells to cause the response of cell death? Are you finding them?
Yeah, I'm trying to. I got a little bit just now, and then it kind of went out of focus.
It's important that we're studying this because carbon black is a component of air pollution, and that's something that affects all large cities around the world. So it's really interesting because it's teaching us more into how exactly this is affecting our bodies, and hopefully in the future this can help with treatment. So what I'm doing right now is I'm preparing the dye that I'm going to put on my cells for an experiment, and this tests for reactive oxygen species. That's a signal that the cells are under stress.
I love that I get to be doing my own thing. I get to decide what I want to be doing with my experiment, and I get to analyze my own data. And while I have Dr. Brewster mentoring me and guiding me through it, it's mostly an independent project. The most exciting part to me is the imaging part, because it's a very beautiful scene. I don't know if other people would find it beautiful, but when you see your cells turn up bright fluorescent blue and then the dying cells turn up fluorescent green, it's a really magnificent sight to see that your experiment's working, and it's just very aesthetically pleasing.
So right now we're seeing here at the beginning the mitochondria are long strings, and that means that they're healthy. Then over time they start to fragment and then circle up into these like a flower bleb kind of shape, and then into round circles. So the results that we're getting here will hopefully lead us to further discoveries in the future. This is one big step, learning that the mitochondria are involved, and it gives us direction into future research, future questions that we can ask, and then what other people who are researching similar topics can ask too.
I was really shocked to be able to get into the lab and start getting hands-on my first semester coming here. When you're taught by a teacher, it seems more straightforward, and you're like, okay, all I'm gonna do is get some gel and put it inside a bottle and then mix it, and then hopefully it turns out right. But once you get all the data and stuff, it becomes more of your own understanding.
These samples are immunoprecipitation samples. So what we do is we stick an antibody onto the bead and use it to isolate a particular protein. So out of all this process, we've got a collection of proteins that are super pure because they've been antibody cleansed, essentially antibody purified. And the beads help us to grab those proteins and isolate them from solution.
Being able to actively do what they're saying and maybe even help them in some form of their research, it really makes me feel more integrated with the teacher, and I'm able to talk to them not as just a teacher, but as more of a student and a friend.
It's important that students get engaged in research their freshman year, because that gives them so much more time to get used to working in a lab, get used to what it's like being a scientist, being a researcher. And then once they learn those skills, they're able to come up with their own individual projects and pursue their own questions.